Hello, in this video I will explain how to create your Proteomics and Transcriptomics pathway analysis visualizations using the R app EnrichViz. EnrichViz app can be downloaded from the GitHub repository using the link on your screen or by following the link on the video description. Once downloaded, it can be opened on RStudio version 4.2 or later and run by clicking on the Run App button. Additionally, EnrichViz can be run directly from the Shiny app website following the link on the screen or the link in the description. To generate your visualizations, the app needs 3 CSV files. The first file is a normalized protein abundance of transcript or normalized transcript data. The second file is a sample annotation file or the metadata file. And the third file contains the results from the enrichment or pathway analysis regardless of the tool you use, as long as it contains a column with pathway or function name and the molecules are overlapped with those in your data. So the app generates 6 main visualizations: a bar plot, a bubble plot, a chord diagram, a heat map for each pathway or downloaded, or you can download heat map for all the pathways at the same time, and a box plot or violin plot for the selected gene or proteins of interest. Future version of the app will add all the visualizations of volcano plots, principal component analysis, among others. I will leave in the description a blurb you can use to write your methods if you use the app, as well as how to cite this tool. So let me show you how easy to generate those visualizations using this app. As I mentioned before, you can go to the GitHub repository website with the link that I will leave in the description and download the app from code, download the zip file, and then unzip it and open it. So once you are in R, you open the application and then you can run the app from this button here, run the app. The app will open and it's going to ask you for those 3 files. The first file is a normalized data where it is normalized abundant or it is a normalized counts if it's transcript data. So you browse that file. Gonna use normalized counts, RNA-seq experiment, and upload it. If you wanna see how that look, it's just a file that contains gene symbols and the values of those normalized count for each of the samples in your experiment. The second file is a metadata or sample annotation file. Browse. Annotation file in this case, if you wanna see how that looks. It's a file that contains unique sample IDs as well as annotation for each of these samples, depending on the group that those samples are in your experiment. You can have one annotation, simple annotation, as I'm showing you. You can have many other annotations for each of those samples, depending on the complexity of your experiment. And the last file that you need is all these files are comma-separated files, as I mentioned. And the last one is the enrichment results. In this case, the RNA-seq enrichment is a G-profiler enrichment analysis results, and you upload that. If you want to see how those look, I'll show you. Basically, it has pathway names here, in this column called term name, and the basic information that you need to use this app is what are those genes are overlap with those pathways or functions. It may have other information that you can use for your visualization, and I will show it in a second. So we uploaded the 3 files. So you can see it says error object because we have to define what are those columns, what are those parameters for the system to use it, the application to use it. And the first one is select the pathway function, and in this case it's called term name, as I show you. See, right away it generates the bar graphic. The molecules in this case are intersections. Okay, it got it right. Otherwise, you can change it and select the column where your molecules are. When I mean molecules, in this case, it's gene symbols. And if your enrichment file is not comma separated, you can have a different separator. You can set as a backslash or semicolon. You can select those. In case of the normalized counts, gene or protein ID is not description, of course, it's called gene symbols, this one. And it's not going to be used for this one specifically, but if you use a core diagram, it's you're going to give you an error if you don't select the right one. In this case, a sample notation is sample ID in this case. Otherwise, you can select depending on how you call it to your samples. And the group that I have in experimental category, I call it group. You can have other names. So you select, if you have different ones, you can select the one that you want to represent. So the second graphic that we're going to use is a bubble plot. So in this case, we have bar plot. You can do bubble plot just by switching from those two. And in this case, notice that we're using adjusted p-value. So raw p-values detected. It applies the minus log 10 transformation so you can visualize the p-value. So the higher the number, the more significant it is. So if it's not, you have a p-value that has been already logarithmized, it says it detects it and it says it's already log transformed. I don't need to do anything else, so it's just going to use it directly. So you can change the number of pathways. Let's say we want to do 10 pathways, and you can say let's do 10. In the term size, you can use a term size is how big is each of the pathways. I don't want to use that. I want to use the intersection size. So how many of my molecules overlap with those pathways, which is something that is going to change. So that's what we are using in this case, the intersection size. The color is we can do adjusted p-value for the color, but you can choose another thing like, let's do intersection size, for example, again. So you can see the color changes depending on this a bigger intersection size or not. So, but you can change any other one. I can say like the p-value, intersection size. Play with those combinations as you can change the color. You can change, for example, yellow to red or red to blue, 3 scale, or you can use a different one. So play with that and select the one that you feel it's best represent your data. So the second visualization is a core diagram, this one. And you can change the top pathways to include. It has 2, but you can, let's say we can do 10. And minimum times a molecule must appear to overlap with a different pathway. We can send to 2, for example. You see less molecules, and you can continue increasing 3 or more. And also you can change the size of the inner circle to reduce the clutter of this. And that's something you can play with that. It's the font size of the pathway. We do 4 or 6. Or you can, so the molecules the same, you can do 0.8, so increase the size of the molecules, etc. And also the color of these bars, or each of the, so in this case it says tomato, so it's for the color for the pathways, so at these ones, the pathways. And you can also you can type the color, you can set the red, dark red, for example, just to show you can change that. But you can also use a hex code. If you're not familiar, you just Google hex color and you will see this web page showing you this. So you can move the cursor here. For example, I want to let's say more like green, and this is the hex color of this dot. I can move it around, see, and darker. I can say, okay, I copy this number here, down here, and I put it on my, instead of typing, I can put the hex color and see how that changes. So you can also use that. And same for the color of the molecules, of the proteins or transcripts. You can change the height of the plot, and when you're happy, you can download, as you can do with the bar plot or bubble plot. You can download your figure. In case of the heat map, this is the other bar here. Click on the heat map. And you can select which of the pathway you want to see how the molecules change across a group. In this case, I'm showing the first one. You can say, let's say metabolism of steroid. You see how the molecules change. So those are mostly down in the control group and up in the treated group. You can change the color of the groups. In this case, control. Let's say you want to control green and color the treated like red. So for example, just an example, and so see how that changes in the colors legend also. Same, you can modify the height, and you can download either the individual map or all the heat maps, all these heat maps that you decided to bring in the app. So it can be downloaded at once, and then you select the one that you, or the ones that you are you want to present. The last type of plot is BioLink plot. So as you can see how it keeps the color that we gave to the experimental groups. So it keeps green for control and red for the treated, and also you can switch from BioLink to box. So each of those is a sample, and the values of the gene that counts of a normalized count for that sample. So you can change the gene. Let's say I want to change this one, and I can change another one. See how that changes. So show you that it works. Sample ApoE. So see how that is different. For example, same, change the size, the height of the plot, and then also you can download the plot when you are satisfied. So this is in case that you're running the app from the RStudio. If you don't care about downloading the app, you can also run the app from the Shiny app website. Just take the link that I will leave in the description and put it on your browser and select the 3 files. In this case, I can show you how an example with protein data, proteomics data. See, this is a normalized abundance for proteins. This is a annotation data for protein, and this is the enrichment analysis. In this case is ingenuity pathway analysis. Once I have my 3 files, I select the pathway. In this case, enrichment is called ingenuity pathway analysis. The molecules is saying the molecules that overlap. It's called molecules. Fine. It's a comma separated file. I don't have to change anything. In case of the normalized counts, the gene symbols are the gene symbols. The sample ID is called sample ID, and the group is called group. But see that there are other annotations. If you can select. I don't want to go over details. I can do also switch to bubble plot, and I can also change the number of pathways. You can change some parameters, have a Z-score instead of the ratio. So you can do the ratio in this case for the size, but you can change, let's say, the Z-score if you care, and also the size of the scale of the bubbles, how they look in the plot. Change the palette, as I already explained. Instead of the in case of the core diagram, you can also change those parameters I explained before. I don't want to go over, but see, it's a data agnostic. You can either put transcriptomics, proteomics, and I'm totally sure you can put metabolomics. I haven't done the test, but you can do that test and let me know how it works. And same as I already explained for the heat map. In this case, heat map, you will see some different heat maps. Let me switch for the heat map tab, and you can say, okay, and I have the heat map. Let's do autophagy, changes to autophagy. Let's do, I don't know, like this other pathway. You see how that changes. Same for the colors of the groups. You can change the colors of the group. I have already explained. And finally, you can do from bubble plot to box plot to BioLink plots, and select the gene of your interest that you want to show. So that's it. That's how it works. So stay tuned for future updates. As I said, I will include the volcano plot and the PCA in the app for your easier visualization of the omics data.